CO.
Okay, thank you for the introduction and thank you all for being here. The work I will present today was made in collaboration with other Argentinian researchers from the Argentinian Museum of Natural Sciences, that's where I work, and 2 Canadian researchers from the Toronto Zoo and the University of Guelph in Canada. Just before I start, I want to mention that the results I will show you today have just been accepted for publication in Journal of Avian Biology. So if I convince you that this is actually interesting, you should definitely check our paper because it will have a more deep and thorough discussion of our results. So back in 2010, when I was writing my PhD project to apply for a fellowship, I started to notice how key was molecular dating in evolutionary biology studies in general, and how common it was to apply the same evolutionary rate across different loci and different mitochondrial genes, different taxonomic groups, sorry. So I started to wonder if that process was actually correct. And that motivated me to perform this large assessment of the evolutionary rate of CO1 in birds relative to that of the Cytochrome B. And I will get to why we chose this CO1. So as many of you will know, most current knowledge of the tempo and mode of avian diversification relies on the estimation of the age of a species and whether the timing obtained is compatible with a particular evolutionary scenario. To date events in the past, one of the most common ways today is to use genetic data through the application of molecular clocks, that is, molecular dating. To do so, we first need to calibrate these molecular clocks using time reference, such as the fossil record or biogeographic or geological data. However, the utility of molecular dating relies on the proper calibration and application of molecular or evolutionary rates, which in the case of birds are currently mostly derived from the mitochondrial genome and particularly from the Cytochrome B sequences. And in this context, I must mention the well-known 2% rule in birds, which has been subject of strong debate on whether it is safe to assume it is constant across different taxonomic groups of birds. So what do we know already about molecular rates in general and on the avian mitochondrial molecular clock in particular? First, we know that there is considerable rate variation across taxa and protein coding loci that could be caused by either gene effects, that is, differences in the absolute rate of different genome regions, or lineage effects, this is disparities in some species traits such as generation time or body size that could cause evolutionary rates to vary across taxa, or by a combination of both effects. Either way, this high variation questions the existence of universal molecular clocks in birds. Secondly, we also know that molecular rate estimates appears to be negatively associated with the age of the calibration the calibration point used to estimate them. This is represented here in these 2 figures. This pattern was reported first by Garcia Moreno in 2004 and later by Hoet and collaborators in 2005. And here you can see how rates estimated within a species or recently diverged lineages are significantly higher than those estimated with older calibration points. So in this context, we decided to perform a large-scale assessment of the evolutionary rate of the CO1 relative to the site B, and we chose this gene because of 3 main reasons. First, there exists already a large and well-curated library of CO1 sequences as a result of the DNA barcoding initiative. Highly tied to the first point is the fact that it has become a frequent choice alone or in combination with other loci in evolutionary biology studies, but many insights it could provide are currently hampered by the lack of a calibration of its evolutionary rate. So we first mined from bulk and GenBank all publicly available CO1 and Cytochrome B, respectively, sequences from 10 avian orders. Then we use published phylogenetic information to generate as many phylogenetically independent pairs of species or comparisons as possible within each of these 10 avian orders, and we compare for these pairs of species the level of divergence in both loci. This resulted in a final dataset of almost 300 pairs of species from 10 avian orders, which appear listed in this table, represented by almost 3,000 CO1 sequences and over 2,000 Cytochrome B sequences. Then we use omega to calculate uncorrected genetic distances between for both loci, and then use these distances to estimate an index that indicates for each pair of species the difference in divergence between the both genes. This index, called the Cytochrome B-CO1 index, I will refer to it throughout my presentation. The important thing here is to keep in mind that positive values will indicate that the distance in cytochrome B is higher than that in CO1, while negative values will indicate the opposite, higher distance in CO1. So before getting into results, I just want to mention two controls that we performed to test for any bias. The first one was intended to test for any bias introduced by the comparison of CO1 and cytochrome B sequences that did not came from the same individual. So to do so, we created what we call the per gene dataset that was like a subset from the total dataset restricted to 150 species, for which we knew for sure that both genes were sequenced from the same individual. The second control was intended to assess the effect of analyzing uncorrected genetic distances, because as you may know, this type of distances could potentially underestimate the true divergence between species due to mutational saturation. We used two different approaches. I will not get in detail into this, because for those who will check our paper, this is a spoiler alert. The patterns obtained in both cases were almost identical to the ones derived from the analysis of the complete dataset and the analysis of uncorrected genetic distances. So trust me, and no bias was introduced by either the comparison of sequences that were not derived from the same individual, or by using uncorrected genetic distances. So, results. We found that CO1 evolves on average 14% slower than cytochrome B, but as you may already have noticed from this table, there is considerable variation in the value of the index among orders. For example, on average, the distance in cytochrome B and CO1 seems to be fairly similar for groups like the strigiformes and the apodiformes, where cytochrome B tends to be significantly higher, considerably higher than CO1 in groups like the anseriformes and the gachiformes, with all the other orders showing intermediate values. Note that in any case, CO1 was found to evolve on average faster than cytochrome B. A closer inspection revealed that the variation was also high within each of the orders, and was also related or associated with the level of divergence between the species that we are comparing. For example, let's look at this table where we have the case example of the gachiformes. Here you have the 8 pairs of species that we use, and here you can see that when the divergence between the species is low, that is closely related species, the value of the index is high. Whereas when the divergence between the species is high, that is distantly related species, the value of the index is low or even negative. That represents higher distance in CO1 than in cytochrome B. So to further investigate this pattern, we compare the divergence values of both loci across the entire dataset, which appear summarized in this scatter plot. Here you have in the y coordinate the divergence in cytochrome B, and in the x-axis you have the divergence in CO1. The dashed line corresponds to the equal rate relationship, the one-to-one relationship. And as you can see from here, there was a significantly high association between the divergences in both loci across the entire dataset, and it was also close to the equal rates relationship. But if we look here in this zone of the scatter plot, the distance for cytochrome B tended to be higher than that for CO1 in the species pair with low CO1 divergences, whereas values tend to equalize as the divergence between the species increases. The influence of the level of divergence between the species gets even more clear when we plot the value of the index for each pair of species as a function of the divergence in CO1 for that pair of species. Here you can see that there is a marked decay of the value of the index as the divergence increases, meaning that genetic distances in cytochrome B tend to be higher than those for CO1 for recently diverged species. Finally, we found that the mean value of the index for each order was strongly and negatively correlated with the mean CO1 divergence between the species pairs that are being compared within each order. That suggests that differences in the value of the index among orders could at least partially be explained by the mean divergence between, or the mean age, between the species that are being compared within each of them. So which are the main conclusions that we can get from our results? First, we have shown that genetic distances in Cytochrome B tend to be higher than those in CO1 for recently diverged species, which cause the marked decay of the index that I have just shown. And this decay of the index does resemble the pattern of time dependency of rate estimates, even though our study does not involve absolute rate estimates, but it is a relative comparison between two loci. This similarity suggests that in our case, this standard effect, that is accelerated short-term rates, might be stronger in Cytochrome B than in CO1, or that CO1 evolutionary rate might be fairly constant. But why are we seeing these differences between the two loci over short evolutionary periods? Well, to start to address that question, we first need to know that prior studies have already shown that CO1 is the least variable gene in the mitochondrial genome. It's the one that shows the less rate variation across lineages, and the one that better fits a clock-like model. Furthermore, non-synonymous substitution in CO1 are significantly lower than for other protein-coding mitochondrial loci, whereas synonymous substitution across the mitochondrial genome seems to be fairly constant. So one possibility is that differing intensities of negative selection pressure could explain the difference observed between these two loci over short evolutionary periods. If Cytochrome B might initially accumulate more mutations more rapidly than CO1 if it possesses some hypervariable sites where substitutions are only weakly exposed to parifine selection, while CO1 lacks such sites. And this is called mutational hotspots. Even though looking for mutational hotspots is really complicated, we perform a quick examination of some of the closely related pairs of species within our dataset, and we found no clear signs of persistent mutational hotspot, that is statistical significant signs, but our results suggested that some hypervariable sites in its sequence may exist. Therefore, a more comprehensive and more thorough analysis of the Cytochrome B sequences, like the one performed by Galtier and collaborator, is needed to better assess the existence of these mutational hotspots in the Cytochrome B. Which are the implications of our results for other evolutionary studies? Well, since Cytochrome B and CO1 behave differently only for recently diverged pairs of species, both loci could be used to estimate the age of a species, because most species do not fall in the danger zone of low divergences. When working in phylogenetic analysis, which cover a wide range of species divergences, CO1 seems to be more appropriate than Cytochrome B because its evolutionary rate seems to be more uniform. On the other hand, this gene is particularly constrained between recently diverse taxa. So if you're working below the species level or trying to gain resolution in ongoing speciation, you should definitely use another gene, but always keeping in mind that patterns like the time dependency pattern could cause a bias in the molecular dating process. And finally, what are the further directions of our work? Well, we believe that it is worth revisiting past calibrations to estimate rates for other genes like CO1, because we believe that assessing if the patterns we report here are sustained when comparing absolute rate estimates together with a deeper investigation into the existence of mutational hotspots in the avian mitochondrial genome could certainly advance our understanding of evolutionary rates and their variation across loci and taxonomic groups. And I want to show you the slide of my acknowledgements, and thank you all for listening.